Thank you very much, Susan. Thank you very much, Shirin, for being here with us today. And thank you, all of you in the audience, for joining for what I think is going to be a very interesting presentation. I'm delighted to introduce to all of you Dr. Shirin Golchi. She joined the Epidemiology and Biostatistics department at McGill only in 2019, and we'd have probably seen her sooner if not for this pandemic. But Shirin brings some very interesting research interests with her, which is why I say we're all excited to hear this talk. Her interests are in the area of Bayesian methods and computational statistics. And she has a particular interest in Bayesian adaptive trial designs, which is what she's going to talk to us about today. She completed her PhD from Simon Fraser University in 2014. And Shirin, I'm sure you've heard this already from other people, but once upon a time in EBOH, there was Lawrence Joseph, who I think who popularized Bayesian statistics, and Stan, I'm sorry, I don't remember his last name, but our other colleague who was an expert at clinical trials. And you seem to be bringing both those expertise together in your own interests. So we're all very keen to hear from you because you're doing something modern and new and taking forward the ideas, the ones they used to talk about.
Thank you so much, Nandini. Thanks for the invitation. It's a pleasure to be here, and thanks for the nice introduction. I have to clarify that I'm sort of posing as a biostatistician. My training was in statistics, and I worked at a health research company for a couple of years, and that's what got me into Bayesian adaptive clinical trials. So I hope that I won't disappoint from that perspective that for my little experience that I've had in this area. Okay, I'm going to start sharing my slides. Okay, can you see them okay? All right. So I changed the title of my talk a little bit. Previously it said Assessment of Design Operating Characteristics in Bayesian Adaptive Clinical Trials, and I changed Bayesian Adaptive to Modern. I will define modern in a little bit. I will still be focusing on Bayesian Adaptive Clinical Trials. So the talk hasn't changed, but I just wanted to emphasize the fact that the methods that I will be talking about can actually be used in a more broad sense in not only in Bayesian adaptive clinical trials, but wherever we can do a closed form sample size calculation. I will talk about in more detail. So I will start with a brief introduction, basically defining modern clinical trials and the family of clinical trials that I'll be focusing on. And then I'll talk a little bit more about Bayesian adaptive clinical trials and provide some examples. I talk about what we currently do when we plan these kind of trials, when we do power analysis, and when we explore the operating characteristics of these kind of trials. And then finally, I'll talk a little bit about the type of methods that I've been working on to make this process a bit more efficient, basically. So when I refer to modern clinical trials, these are basically, these are trials as opposed to the traditional randomized clinical trials, RCTs, that we're all very familiar with. They've been known as the gold standard for many years. And it's basically the simplest design where we are randomizing patients into two arms, a control and an intervention arm. And ahead of at the planning stage, we basically calculate these sample sizes that are required to detect a given effect size and assumed effect size. And when we design the trial, when we calculate the sample size, then we commit to it. We basically want to complete the trial to achieve that sample size, and we won't look at the data before that. And when I say continue to the end, no matter what, of course, it doesn't mean that we won't be doing safety monitoring. Of course, there are all sorts of monitoring processes in place to make sure that the trial is safe to the participants. But from a statistical perspective, we're not supposed to to stop or look at the data or do any kind of analysis before getting to the end. So that's what we mean by fixed sample size. And in principle, because we're doing randomization, we don't need to adjust for covariates or do any kind of complex analysis. It could be as simple as a t-test. Again, in principle, in practice, and we will have to sort of think about our analysis a lot more carefully in specific contexts. But I'm also, when I refer to modern clinical trials, I'm also not thinking about some kind of more advanced designs that could that are in place rather than the traditional RCT, the parallel design, such as cluster randomized trials that might be used for logistic reasons, or group sequential designs where we are actually looking at the data before at interim analysis before the final analysis. But so I'm excluding these designs because we have developed some kind of statistical theory or approximations to be able to still perform closed form sample size calculation and power analysis for these. So when I refer to modern clinical trials, it's the case where we are going with a complex enough design where we don't have the statistical theory to be able to do closed form sample size calculation. So as I mentioned, I will be mainly focusing on Bayesian adaptive clinical trials, which I'm going to think of as a specific category of these modern clinical trials. But I want to emphasize that the methods that I'll be talking about are not restricted to these family of trials. So adaptive means that we have these predefined adjustments that we plan ahead of time, that we are allowed to make to the design at pre-planned interim analysis. And again, I'm going to emphasize that these are planned adjustments. So we define our decision rules very clearly ahead of time. It doesn't mean that we can make any kind of ad hoc adjustments as we go through the course of the trials. These adaptations could be adapting the allocation ratios, the randomization ratios. For example, if a treatment happens to be working, if we have enough evidence that a treatment is working better than the rest of the treatments in a multi-arm clinical trial, for example, we can basically adjust the randomization ratios such that we allocate more patients to the more effective treatments. We could stop the trial at a certain interim analysis. We could stop the trial for efficacy if we have sufficient evidence that one of the treatment arms is superior to the rest. We can stop an arm or the trial for futility. Let's say if we have enough evidence again that the treatment is ineffective, we can basically stop randomizing patients into this arm, or we can stop the trial altogether for futility if basically at some point we realize that the effect sizes that we assumed aren't there and the trial is hopeless. We can basically reconsider our sample size estimates if we have more information about our effect sizes, or we can prolong the study to give us a better chance for detecting the effect sizes if it's possible. And these kind of adjustments are made for different reasons, for two main reasons. Ethical reasons, as I mentioned, we can basically reduce the number of patients who are assigned to an ineffective treatment, or let more patients to benefit from an effective treatment, or for cost reasons, because when we allow for stopping early and making these kind of adjustments, we often end up safe in terms of sample size or even the duration of the trial. So our expected sample size will be smaller in many cases when we're using these kind of adaptive designs. So why Bayesian? I would say the main reason is convenience. Because we are thinking when we are thinking in terms of adaptations, in terms of having multiple interim analysis, we're basically thinking about these sequentially updated results. We are hoping to be able to update the analysis results that we've had from the previous interim analysis using the data that has been collected most recently. And the Bayesian framework basically facilitates this sequential updating in a very natural way, that basically our analysis result from every step is considered as a posterior distribution that summarizes our knowledge about the unknowns of the model, about our effect sizes and the unknown parameters of the model. And then we're updating, this becomes our prior knowledge for the next step, that we are basically updating according to the data that has been collected between the two interim looks. Also, the validity of Bayesian probability statements. So I will be talking about these probability statements that are the basis of inference, the basis of decision-making in Bayesian adaptive trials, and the validity of these probability statements are unchanged, are unaffected by multiple testing in multi-arm clinical trials or by small sample sizes. Again, in frequent statistics, we are basically relying on asymptotic results that assume large sample sizes. But when we are basically calculating the posterior probability, that probability statement is not affected by the sample size based on which we have estimated it. So that's another appealing feature of Bayesian framework. So some of the examples of Bayesian adaptive trials that I've been involved in, one of them is the referred to as the sepsis trial. That's exploring symbiotics for early prevention of severe infection in infants. It's basically a cluster of studies, a cluster of observational studies, as well as clinical trials. It's a large collaboration between the team of researchers at the Sick Kids Hospital in Toronto, as well as a large team in Bangladesh, based in Bangladesh, and these studies are being conducted there. And the study is sponsored by the Bill and Melinda Gates Foundation. Where I come in is basically the design of the phase 3 clinical trial that looks at right now we're looking at the 2-arm clinical trial, basically comparing the placebo and a specific type of symbiotic L. plantera. But as I mentioned, this is basically a cluster of studies. They're also looking at a sort of a dose funding study that looks at another type of symbiotic. And at some point, they might, depending on the result of this dose finding study, this other symbiotic might be added to the trial, or a combination of these two might be considered. That this is basically a sort of an imaginary picture, but this is just to emphasize that there are a lot of flexibilities that are being considered, and this could be sort of fall under the platform clinical trials where you're allowed to sort of drop or add arms along the way. What makes the design that we're considering right now very interesting is that there are different definitions for sepsis that sort of vary in terms of stringency. So, for example, we can define sepsis as non-injury related death or blood culture confirmed sepsis. These are all composite endpoints, by the way. And, or we can define it according to sort of community health worker diagnosis. And what's interesting about these endpoints is that they have this hierarchical nested structure. That's something that's considered a case of sepsis under the most stringent definition is also a case under the permissive ones, but it's not correct the other way around. This becomes very, this is very important when we are simulating such a trial to incorporate these outcomes. Now, when we have these endpoints available, it's the question is basically which one is the most appropriate using in a clinical trial. Specifically from a statistical perspective, it could make quite a bit of difference in terms of power, because you can imagine that the base infection rate or base rate of sepsis could be very different under these different definitions. And if there is an effect size with a larger base rate, of course, depending also on the mechanism of the effect, it could make quite a bit of difference if we are using a rare an endpoint defined based on rare events rather than one that's defined based on more frequent events. So what we are exploring right now is that can we monitor all these endpoints, and can we define stopping rules that are also composite stopping rules that basically rely on a combination of these outcomes. For example, it can be defined as superiority stopping rules with respect to the most stringent definition of sepsis, but stop the trial if we have sufficient evidence for futility with respect to the more permissive outcomes. And so there are a variety of stopping rules that you can define based on these combinations, you can imagine. And there are also other design parameters that we will have to explore. For example, we'll have to look at the number and spacing of the interim analysis. We'll have to look at assumed effect sizes as well as assumed base rates for each of these definitions, etc. That will give us basically a large number of scenarios that we will have to explore when we're looking at the design operating characteristics of a clinical trial with this kind of structure. Another more recent example was this trial. It was a COVID trial, basically focusing on prevention of a severe case of COVID using high-dose vitamin D in healthcare workers. Unfortunately, the trial didn't go through due to enrollment issues, but I still find the design quite interesting. I think it's worth going over. The pandemic actually brought Bayesian adaptive designs to the center stage. And I think the most relevant example is the Pfizer vaccine trial that was using a Bayesian adaptive design. And here it's the reason why these kind of designs became very popular for COVID trials is that we're in a situation where we want to conclude our trials as soon as possible. Basically, everyone is looking for results as quickly as possible. There's also competition for patient populations. So we're basically, we would like to keep our sample sizes as small as possible in addition to concluding as early as possible. So the other aspect that makes these kind of designs appealing is that we have these rate of infection that's varying quite a bit. So it makes quite a bit of difference if you're during this period where the number of cases is increasing, let's say at the beginning of the second or third wave, or when you're starting the trial where we were during those, sort of a flat area during the summer where we didn't have that many cases. And that makes quite a bit of difference when you're in terms of power, in terms of when you're starting your trial. So what was basically planned for the design of this trial was to look at have an interim analysis when we have 75% of the participants' data available to us, and basically sort of monitor the infection rate throughout the trial. We're going to stop for efficacy if we had evidence, which evidence was basically measured by the posterior probability that the odds ratio that the vitamin D is effective given the data crosses an upper probability threshold. And then, as I mentioned, we would be monitoring the infection rate. And let's say if our interim analysis happened to be around the 12 weeks, and we realized we would basically look at our chances for the end of the trial that based on how the infection rate was evolving, based on all the data we could get from the general population, et cetera, to sort of predict this trajectory and be able to see if we will have sufficient power to continue the trial, or do we have to continue more depending on if the number of cases was rising or basically decreasing. So this is basically just to give you an idea for the different contexts where these kind of designs could be useful and popular. But I will be talking about what it actually takes when it comes to planning these kind of trials. What do we actually do when we want to calculate sample size and sort of provide the basics of the trial design, what everyone is looking for, basically, to be able to get things started and estimate the budget, etc. So sort of going back to how do we do sample size calculation in general when it comes to clinical trials? We basically have these test statistics let's say in the simplest case under an RCT, this test statistic could be a T statistic. And we're basically powered, the definition of power is the probability that this test statistic crosses a certain pre-specified threshold, given that assumed effect size, the chance that we detect this effect size is actually at least 80%. And then we're looking for the sample size that will guarantee us at least 80% power, at least 80% chance that our test statistic is crossing this threshold, given that the effect is actually there. And false positive rate, of course, is the same thing, but assuming that there is no effect. So the probability that we falsely declare that there is an effect, and that is equivalent to the case that our test statistic is greater than this threshold. So all of this is actually based on the sampling distribution of our test statistic under the null hypothesis. That is, what is the distribution if I, let's say, generate data over and over again? If I repeat my trial over and over again, what is the distribution of this test statistic if there's no effect? And then I compare my actual observed test statistic with thresholds of that distribution. So that distribution is basically the key object in sample size calculation. If I have that distribution, then I can do sample size calculation in closed form. If I don't have theory or approximation for that sampling distribution, then all of this basically breaks down. Now, what has changed in Bayesian adaptive clinical trials is that the definition of power is the same. There is sort of a bigger argument that if you're using Bayesian methods, maybe you shouldn't be relying on a frequentist operating characteristics. Maybe you shouldn't be looking at power at all. But of course, we have a long way before we can convince our regulatory agencies that that's not what you should be looking at. So we will have to still provide power and false positive rate and all the frequentist measures that everyone understands and cares about. So the definition of power remains the same under a Bayesian adaptive clinical trial. What changes is this test statistic. This is not the t-test anymore. We're looking at a posterior probability. We're looking at posterior probability of effectiveness in this case, and we don't have any statistical theory for the sampling distribution of this posterior probability. So I don't have statistical results that tell me if you repeat the trial over and over again, what would be the distribution of this posterior probability. And that's why what we're relying on currently are simulations. So what we do is that we simulate our trials on the computer, we generate data from a clinical trial for a given design with all the adaptive features, with all the complexities. And when we generate data for clinical trials over and over again, we generate this posterior probability over and over again and look at the distribution. And that basically, we estimate the distribution using Monte Carlo sampling. And that's how we go about estimating power for a given trial design for a given sample size and do our sample size estimation in that manner. What makes it challenging is that often there are too many free parameters. So I'm going to start from the bottom of this slide. There are the model parameters, and that is the effect sizes and the baseline measures, the infection rate, for example, in the COVID example, that of course we don't know. We have to do our sample size calculation based on an assumed value of these parameters, but it's our uncertainty about these is the main reason why we are even conducting the clinical trial. So there's uncertainty about these, and we often want to explore our operating characteristics for a variety of effect sizes, for a variety of infection rates, etc. So there are a variety of these. So that basically defines a set of simulation scenarios. And in addition to that, we would like to explore a number of design parameters. For example, what would be a good threshold for this, an appropriate threshold for this probability of posterior probability of effectiveness? How high should this probability be so that if I conclude efficacy with respect to this threshold, I'm actually meeting that 80% power? And the same for futility. What would be a low enough, like if I'm defining my a futility conclusion based on this probability of effectiveness falling below a lower threshold, how low should this threshold be? Basically, these are the design parameters that we would also want to explore in our simulations. How fast should we adapt? How soon should we start to adapt if we are basically adjusting our allocation probabilities? Or how often should we look at the data? So these are all the different aspects of the design we would like to explore in our simulations. And finally, there are different sample sizes under the Bayesian adaptive framework, because we are thinking the actual trial sample size is now a random number because we're allowing for early stopping. Things that we need to decide on are the interim sample sizes, basically, what are the sizes of the batches of data that we are collecting between every 2 looks, and a burn-in sample size, which is basically an initial sample size that we want to collect before we even start adjusting, making any changes to our design, etc. And of course, a maximum affordable sample size, because let's say neither of our stopping rules have been satisfied, how far can we go? Basically, our budget should specify when we need to stop, when we weren't able to stop with respect to any of our stopping rules. So these are basically a variety of parameters that we need to specify. Okay, so this brings me to this web app that we developed where I worked before coming to McGill. It was a health research company. It was basically a startup sponsored by the Gates Foundation. And we provided statistical consulting for the design of Bayesian adaptive trials to the foundation. And this web app basically, so this is the address where you can get the web app. It's actually publicly available, but I'll be demonstrating with a local version of it that is slightly different on my computer, but it's basically has the same functionality. Okay, so what we have here basically is the design parameters that I was just talking about, in as well as model parameters. So you can specify your outcome type. I have added some completely arbitrary effect sizes here. There are various aspects of it. I'm not really focusing on the app, but I just want to sort of deliver the message that what it takes when we are thinking about performing simulations for these kind of designs. So there are basically a variety of things that you can change about the different features of the design. You can use a platform design, which means that it will add your final treatment later on to the design. You can compare all the arms simultaneously or compare them to a common control. You can adjust your futility and superiority thresholds. These are those probability thresholds that I just mentioned. And you can sort of decide if you actually want to change the randomization probabilities with respect to the performance of the treatment arms or not. And finally, there are the sample sizes, the ones that I just listed, and a cost variable, which was very important whenever we were talking to people who are sponsoring these trials. That's although the way this is built in the app is, this is actually $1 per patient. So it would be exactly equivalent to your sample size, but it's somehow easier to communicate when you actually put the $ in front of it. So these are basically just some kind of hypothetical scenarios. I'm just going to hit run. This will simulate 100 of these clinical trials with these specifications, and will give power and false positive rate estimates. You can look at the distribution of the final sample size throughout these 100 trials. So these are basically, the height of these bars would be the proportion of times. So we had 4 interim analyses with the sample sizes that we specified. And this shows the proportion of times basically that the trial stopped at the first interim analysis, second, third, fourth, and the final analysis. And we'll make some comparisons with an RCT. And you can also get a sense of your design by simulating single clinical trials. For example, this is allowing for arms to stop, and sometimes the first arm, which is basically has like 0. If you assume that this is a response mean that has 0 response mean, sort of assuming that that's a control or reference arm. Anyways, so the point I want to make is that this is basically just one setting. And think about all the possible scenarios that could arise as a result of considering a range of possible values for each of these settings. That would basically result in a large number of scenarios that you will have to explore, and you will have to wait maybe the number of seconds that we waited for it for these simple designs. Also, the outcome types that I'm considering here are very limited. I'm looking at a continuous outcome and proportions. Count outcomes could be also realistically added, and maybe survival time to event outcomes, and with simple models. But what I'm trying to say is that the models, the statistical models that are used under this web app are very, very simple. So even now that I have a much bigger task in front of me for sample size calculation with simulations, these are still very simple scenarios. So this is possible. This is not the easiest, but it's possible. But we will get to cases where even this is not enough, basically. All right, I'm going to go back to the slides. All right. Okay, so what are those cases where this is not going to be feasible anymore, realistically, when it comes to trial planning? So in these, in the web app, and in most of the models that we are currently using under Bayesian adaptive trials, we're assuming that these updates, the sequential updates that I've been talking about, can be done analytically. And if you've come across Bayesian analysis or talked to people who use Bayesian analysis, you've probably heard of conjugate priors. And this is the case where we have this modeling framework where we can assume a prior distribution, where we can actually, where the posterior distribution combined, when we combine it with the data, the posterior distribution is from the same family of distribution. So we can analytically basically update the parameters of this distribution according to the most recently observed data. So everything is done mathematically. Now, in most realistic cases, or in most interesting Bayesian models, we actually don't have a closed form posterior. So we will have to use approximations or Markov chain Monte Carlo sampling to be able to draw samples from these posterior distributions, and that's what the inference relies on. So what that means is that every time we want to update our results, we are actually relying on more simulations. So this basically adds up to the computation. Every time I simulate a trial, within this trial simulation, I'm doing these updates that also require simulations, which adds up to a lot of computation. Now, we don't have to go very far or make it very complicated to come across one of these examples. I'm sure you've heard about the ordinal scale disease progression endpoint that was very popular in COVID trials. I'm sorry, I'm looking at your faces, which I put on my other screen, so I can imagine that would give a weird angle to my face. So this was the endpoint that was actually introduced by WHO. It was a 10-level ordinal scale endpoint. And here I have a simplified version of it that is basically, it was popular because we were all interested in interventions that could reduce the risk of severe disease. That was basically one of the main interests in clinical trials for COVID-19. So this is basically a simplified version of it, combining some of those 10 categories and sort of reducing it to uninfected, mild, moderate, severe, and dead. And the model, the statistical model that comes with this, that's very popular and common with this type of endpoint, basically assumes that the endpoint of a single patient follows a multinomial distribution, meaning that the patient's endpoint would be in one of those categories with the corresponding probabilities. And then the probability of this endpoint being greater than K, meaning that it is actually the probability of severe disease in a sense, given the intervention they receive. So A represents the arm assignment in a clinical trial context. That's basically just modeled as a logistic regression. So the probability of severe disease is given to us by a logistic regression that has the treatment built into it. And the quantity of interest would be the odds ratio, the ratio of the odds of severe disease for a person who received the treatment to one who did not. So it turns out that for this endpoint and this model, the proportional odds model, that's not a very complicated model at all, we can't actually obtain the posterior probability that the OR, the odds ratio is less than 1 given the data analytically. So we actually rely on MCMC and simulations to be able to calculate this probability. So for example, if we are defining success in such a trial as the case that this probability crosses a higher threshold of 95%, is at least 95%, then the type of questions that we would want to answer during trial planning is that, well, how many patients do we need to enroll to have 80% chance of success, where success is defined as this probability being at least 95%, where the true OR is 0.75 and the base risk for infected individuals associated with each level of this ordinal scale outcome is given as these numbers. And this is a question that's not so easily answered. So what does it take actually to answer such a question is that for a given trial size, we will have to repeat this following process. We will simulate the trial and generate a set of endpoints and calculate this probability using simulations, and assuming that there aren't multiple interim analysis, so there's just one analysis. We're just calculating this once, and then compare the proportion of times that this probability is greater than 95% with 80%. If it's greater, that's great. Maybe we can reduce our sample size and repeat. If it's not even 80%, then we'll have to increase our sample size and repeat. So we have this optimization problem where we don't have a closed form function that we can optimize. We have to just basically plug in numbers, run it, and running it actually takes a long time. And this is we're not just answering that question. Let's say I do this and it takes me maybe a day, and I go back to my collaborators with an answer, and then they say, well, what about an odds ratio of 0.65 and maybe a range from 0.65 to 0.95 because we actually don't know. And what if we want to explore other base rates, basically for base probabilities for each of these categories of this outcome. You can imagine that this problem is basically sort of exploding computationally. And what I'll have to say is that, well, I'll get back to you next week, maybe with an answer, which is not an acceptable answer in the context of COVID trials, or maybe in the context of any trial. So, this is actually a web page by Frank Harrell and his colleague, and they basically discuss this outcome and the PO model and what it takes to actually design a Bayesian adaptive clinical trials, which inspired me in the first place to look at this problem. So now I will briefly talk about the type of methods that I've been working on to sort of provide a solution to this problem. So suppose that you don't run simulations for all of those scenarios, but maybe for a subset of those scenarios that's affordable. And maybe we can use statistical methods to sort of fill in the blanks. Now here in this plot, we're sort of looking at a two-dimensional subspace of our parameter space, because our vector of base probabilities is a vector of size 4. We're looking at the first component and the odds ratio. And for some values of odds ratios in this base probability, I'm simulating the trial. And what I'm looking at is probability of concluding superiority that you can think of as power, basically. And can I use a method to basically obtain an estimate of my operating characteristics, or actually the sampling distribution of my test statistic from these simulations for the rest of the parameter space without having to simulate the trial again? So I have one slide on my methods, just to make sure I don't get too technical. The idea is that we simulate the sampling distribution of our test statistic at these given points where those square dots were, at a set of initial set of values for our effect sizes and our base risk. And then we learn or emulate or predict the sampling distribution for the rest of the space. And different terminology could be used depending on what field these methods are coming from. And the idea is to use this spatial correlation. What this means is that let's say for effect sizes that are closer to each other, we expect the sampling distribution to be more similar as opposed to parameter values that are further apart from each other. And we are using this kind of correlation to be able to estimate the sampling distribution where we haven't simulated the trial. So we are borrowing techniques from spatial statistics as well as computer experiments. And I'm just going to briefly mention Gaussian process models. These are basically the models that I'm using. They're very flexible models where you can incorporate spatial structure spatial correlation into your model and take advantage of the Gaussian form, that would be very convenient. So that's basically what's being done. So when I do this, what I get is basically these estimates of the power together with uncertainty estimates. So the square dots are what we had before. Those are the estimates of power that I've actually obtained by just simulating the trial, the same way we've been doing so far. And the round dots together with the error bars are my estimates that I've obtained using the techniques that I just talked about for power together with uncertainty estimates. And this is actually the first time we're seeing uncertainty estimates around operating characteristics of a clinical trial. This is not common, but it's of course useful because we are never certain. So it's useful to quantify our uncertainty about these estimates. Now, what's nice about this is that it's flexible. Instead of estimating a single operating characteristic, I'm actually estimating the sampling distribution of my test statistic. I'm trying to get the same toolkits that we have under the frequentist framework. So now that I have the sampling distribution predicted all for a variety of parameter space, I can actually explore different decision rules. What if I define success as this posterior probability of effectiveness being greater than 90% instead of 95% or 98% to be more conservative? And I can basically immediately get these estimate power based on these decision rules from what I have without having to do everything again, like repeat the computation again. And the same way with futility. Now, these are just example estimates. For example, if I define a more relaxed success rule, success criterion, then I will have increased power, for example, for and compared to this plot up here. Or if I make my success criteria more strict, I will get lower estimates of power. And the same with futility. I've restricted this to effect sizes basically where we even have a chance of futility, because futility makes sense when the chance of stopping for futility becomes non-negligible when you're looking at smaller effect sizes. Those are the cases that you would actually want to stop for futility. All right, so skipping these detail. So what kind of difference does this make in practice? This would be an example conversation that I could have now with my collaborators if we are using this model and this outcome, that I basically report the power to them for a given, let's say, sample size of 1,000, assuming a base risk vector of such and such, odds ratio of 0.75, a certain superiority decision threshold and futility decision threshold. I would say that you have 42% to 55% chance of stopping the trial for superiority and practically 0 chance of stopping for futility. You wouldn't even want to stop for futility if you have an odds ratio of 0.75. And if they ask me, what about a larger effect size? I don't have to get back to them in a week. I can say, just a sec, I'll grab that for you. And that would be immediately available by the results that I've already obtained. And of course, they can also explore different decision rules. So it makes the process a lot more feasible as opposed to what we currently have available to us. To conclude, these kind of methods could basically facilitate design of these kind of trials with a lot more flexible and realistic statistical models that would basically capture the reality. We can incorporate covariates if we have to in our clinical trial designs, or we can basically come up with models that are a lot more complicated. And it enables uncertainty quantification, that if when I say that you will have 60% power to my collaborator, that might not be very realistic, but providing a range to them makes me feel a lot more comfortable with the information that I'm providing. And all of this will basically remove the barriers that we currently have in terms of adopting these kind of more complex designs that might need very specialized statistical expertise. So sort of trying to promoting these kind of methods will help us promote these kind of designs to be adopted in practice. And I believe this is just the beginning. There's a lot more room for improvement and development, making these more accessible, more easily, more user-friendly, and hopefully be able to use these designs more in practice and be able to learn more from smaller clinical trials. These are a number of references that I used, and thank you so much for listening. I'm happy to answer any questions you may have.
Thank you very much, Shirin. And to the audience, do any of you have any questions for her?
Yes.
Well, really
Lisa.
nice having you here, Shirin. Oh, sorry, Nandini was.
Oh, just Kabira, please.
Okay. Wow. I'm very happy you're here at McGill now. We'll all come knocking on your door. I feel like we're now going to need a grant in order to plan a trial, because this looks fairly complicated. Just by pure coincidence at one of the journal clubs I attend, we were looking at the RECAP trial, looking at ICU patients hospitalized for severe pneumonia in the ICU, and they have it's an adaptive platform Bayesian approach, and they were looking at these IL-6 inhibitors. Not really important, but it was this kind of design. And they looked at two different drugs. And what I couldn't wrap my mind around, so first you read it and you understand that there's a lot of different assumptions going on, and it's really a lot to sort of take in as an epidemiologist and clinician, because you can't explicitly sort of see it in the summary anyway of the study, the protocol. But then they basically, they tested the first agent, and they saw that it was effective. It reached an odds ratio of 1.77, for benefit. And then the next drug, the people that were recruited in, it was a much smaller sample. And if you looked at their characteristics in a kind of a table one way, it was kind of a different group of people. They were different in age, they were different in SES, and yet they concluded, yeah, this one is also effective. So I didn't know how to take that in. Presumably I could sort of believe that somehow with all of these different frequency distributions, they concluded that giving the way this one was going, that it was probably also effective. But there was a part of me that said, but it's kind of, they're looking at a different constellation of characteristics. So can you make that conclusion? I mean, is this kind of turning over all of our kind of basic epi principles, in a clinical trial, you're randomizing so the two groups are similar, and all of that. So I'm just curious.
I would say no. I don't think this has got anything to do with that, and we can't really replace those with these, like, just because we're using a modern design, that those questions don't go away. So you will still have to make sure that the patient population that you've captured is the one that you've defined for your clinical trial. And I think that's a completely separate question, regardless of the design that you're using. I think that just remains an important question. Something that would be relevant, though, that could be answered from a statistical perspective. I think that you could actually incorporate covariates if you have methods to deal with. I don't know, if you at the planning stage, you could sort of think about different covariate distributions and then incorporate that into your analysis and your power calculation. Maybe you are taking that into account at the planning stage. I don't actually know how exactly to do that, because you will have to think about all the distribution, all the possible distribution, all the distribution of the covariate of the patient characteristics now, and then incorporate that into your simulations, which would add a lot more randomness. So that's one way of maybe to address it. That we could say that these kind of these kind of methods could be advantages, but I would say a very firm no, that you can't stop thinking about those things, because we're using a modern design. So I don't know if that's a helpful answer. But.
No, it is, because at some point you just start wondering, is that, are you just using a bit of data to really populate what is effectively a simulation study? Like, is it a trial anymore at that point? but I'll let other people ask questions.
Thank you.
I noticed that Teresa, I believe, had a question.
Thank you, Shirin. I was so looking forward to your talk because I wanted to learn more about clinical trials. It made me uneasy, changing what I see as essential points of a clinical trial over time. It made me uneasy about doing that. And I wondered about even the participants in a trial and how they would react to this type of modern approach to a clinical trial. I guess it doesn't have any real influence in your thinking about modern trials. And I also wondered about stacking the deck, using the data that you're collecting to influence your own future trial. And that made me uneasy. So I don't know if you could address some of those concerns.
I understand that, and I don't think you're alone in feeling uneasy about it. But the answer is that these are not changes that we just make on the go. These are changes that have been planned. And so all the simulations are meant to sort of know what are the consequences of making these adaptations ahead of time. That's why we have to basically simulate the trial over and over again under different scenarios to make sure that we have thought about all the possible scenarios that could come as a result of making these adjustments. In terms of participants, they will be, of course, informed that this is the design, this is what we are going to do. And in practice, I mean, I know I talked about many different adjustments that you can make. In practice, we are actually being very, very cautious. And I think stopping early is probably the most common version of it that are being used in Bayesian adaptive trials, to be able to sort of stopping ourselves from going forever if we have enough evidence in favor of either conclusion. So those are the most popular ones, and those are the ones that are a lot easier to sort of convince both investigators and participants that this is safe. And this is not going to go wild. In terms of changing allocation proportions, that's a little bit, it's a bit, well, I would say I haven't actually been in a clinical, like in the design of an actual clinical trials where people were okay with that. But I mean, it's the nice thing about being a statistician is that on the computer, everything is possible. So we explore everything. But yeah, I completely understand that in practice, we might be thinking about a lot more limited designs, or honestly, I don't have that extensive of an experience with actual planning of clinical trials to be able to answer these questions, but that's my
Yeah, thank you so much. But we
impression.
absolutely need research in this area, and the COVID situation has certainly brought it to the fore, where we need to be able to have these type of adaptive study designs to get data in a short period of time.
Thanks.
Thank you. I'm going to indulge myself with a question before asking, and I'll let another member of the audience. I was involved in a adaptive planning an adaptive trial recently with Ines Colmenia, who's also in the audience, and Yang Lu, who's a master's student at McGill. And yeah, it was quite an adventure. And I'm looking at your app, and I'm just wondering, could it have saved us a lot of time? All the hours that we put into programming, could we have just gone and used your app? And I guess my question is, how general is that app?
It's a very good question. So to answer your question, we didn't use it ourselves when we were at EPTech. So this is mostly, the app basically serves better for communication. If you're basically just exploring single scenarios with your team of investigators, or you have, the non-statistician portion of your team actually want to explore this themselves. They actually want to try these. And it has, I think you could actually explore several scenarios and download it in a table, but it's not as efficient as if you code your simulations and then you plug in all the scenarios you would like to run the simulation for. So it's a sequential. You can't really do parallel simulation or anything like that. So you were probably better off coding your own simulations. But the purpose of a web app like this, which is very accessible and sort of user-friendly, is for the sake of communication. But it's not that general. I mean, it's basically a beta binomial and a Gaussian posterior. That's all it has, and there's no, like, there are very little things you can actually change about the design.
No, nonetheless, I think thank you. That's very, it's nonetheless very inspiring. So we'll certainly go and look at your app, because you know, even designing such an app is, I know, not a simple matter.
Thank you.
Any other questions for Shirin? Benjamin.
Thanks. That was a really nice talk, and it was well, at least for me, it was geared right to my level of statistical detail versus comprehensibility. I had two questions. One, you may not know the answer, but how is, like, at a CIHR level or other sort of major funding agency level, do you have a sense of how these, like the Bayesian adaptive sort of strategy versus the more traditional frequentist, I guess, rigid strategy, how it's received? Because I could see how, from an efficiency point of view, it's very appealing, so they might like it. But from a not having a sense of the, you know, I guess, like not having a lot of preliminary data to inform the, you know, rates of attrition or the signal or the expected effect size, you might lean on a Bayesian adaptive approach because you don't have that rigorous preliminary data. So is it viewed as a strength because it's efficient, or as a weakness because you're maybe doing a trial when you don't have enough sort of preliminary data? Does that make sense, the question?
Yes, that's a great question. And the short answer would be that it's received much better than before, okay. Especially with, I think now that we have the need for it. So FDA actually has generated one of these guides for industry that just specifically for the design of Bayesian adaptive trials. So they're, they have very specific guidelines that if you're designing a Bayesian adaptive trial as opposed to a fixed design, these are the things you have to do for the FDA to approve it, basically. And so the COVID trial that unfortunately didn't go through, we actually got a CIHR grant for it. So that also went through quite well. But it was one of those rapid, I don't know what it was called, but with CIHR, the program that CIHR put together for rapid response to COVID. So as long as you do we provide the standard requirements that they want for clinical trials, they're actually open to having these flexible features of the design. So as long as we provide a sample size calculation that satisfies 80% power and 5% false positive rate, then I think that would be a check. And from a statistical point of view, I believe. Maybe Nandini has more experience with how this goes in practice, but like with my limited experience, I think the regulatory agencies are a lot more open towards these designs now than before.
Okay,
thanks,
thank you.
Shirin. I agree with what Shirin said, and I know you have a second question. I'll just add here that yes, I think they're really looking for the simulations. We submitted a version without simulations and we got hammered. So yes, you must submit those simulations and show that you have the desired type 1 and type 2 error. And the other question, I think, I don't know who asked that, about the past information. We were advised to use mixture priors, priors that will allow only some weight for the past information, so that your past information is not overly influencing your planning. So I think if those two things are addressed, then at least the statistics part is okay, but then of course they can find fault with other things.
My other question, if I may, it's
yes.
So sometimes we want to, like, I get early stopping because of, I guess, efficacy, superiority, whatever, has obvious rationale for patients and ethics. Sometimes we also want to just know we want to estimate the effect of something. And so I've read sometimes at early stoppage, it can diminish the certainty of the true effect of estimation of the true effect of something. Maybe I have it wrong, but does your Bayesian approach, it always, I guess, requires some pre-specified effect of interest, and so it's not amenable to just estimating the true effect of something, the empirical effect of something. Maybe we can take this offline. And this is probably too tangential. I'm sorry. So I'll maybe send you an email.
Sure. Or if we have time, I'm happy. I'm actually like, that's a very interesting point. And I would definitely like to have a conversation about it. I'm not sure I understand the last part of your question, but is it the true effect or, and
they often will stop a drug.
Okay.
Sorry, I'll take it offline. I'm sorry, because we're past 5 o'clock, but thanks so much. That was a really good time.
No problem. Thank you.
I'd like to just stop and thank Shirin formally. It was a great talk, and so many people have been asking about Bayesian adaptive trials. I think there's going to be many more of them coming, and so I'm delighted that you accepted our invitation and come and shared your ideas here, and we all now know where to go if you have questions in this area. So thank you very much.